Now we're going to talk about genetic engineering. This is when a gene from one organism is placed into another organism. For example, those glow-in-the-dark fish that you can buy at Walmart, those are transgenic organisms. They took a gene from a jellyfish that glows in the dark and placed that gene into the fish. The fish now glow in the dark, which is pretty cool. We're going to look at an example of a human gene placed into a bacteria. This is how insulin is made. They took a gene that made insulin, placed it in a bacteria, and now the bacteria produces human insulin. This insulin is then collected, and that's what they sell when they sell insulin to people who are diabetic. It's actually made by a bacteria in the lab. It's even better than the insulin that was produced by, say, horses or cows, because that's not identical to human insulin. It's close, but some people had allergic reactions to that. When it's human insulin, there are far fewer problems because that's just like what your body would make. So first, the gene that you want is removed from the human cell. In other words, the gene that makes insulin is removed. When we say removed, enzymes are used to cut the DNA where you want to cut it and get just that gene from all of the DNA in the cell. Then you get a bacteria and remove the bacterial plasmid, which is a ring of DNA. It turns out that bacteria have the regular chromatin, uncoiled DNA that you're used to seeing, plus they have little rings of DNA called plasmids. So a plasmid is removed from the bacteria and cut open with enzymes. Then the gene that produces insulin in humans is added to the plasmid from the bacteria, creating recombinant DNA, recombined DNA. Then the bacteria is caused to take the plasmid back into it, and then the bacteria is allowed to replicate. All the new bacteria that are produced from this original bacteria will then have the same DNA and will now produce insulin. It's a lot cheaper to raise bacteria than it is to raise horses and cows. So the insulin that's made using the bacteria is cheaper and more similar to human insulin. How do you cut a plasmid open, and how do you cut DNA and just get out the gene you want? You use enzymes. The enzymes will look for a particular pattern in the DNA. When it sees that pattern, it will cut it there. So every time that pattern shows up, that was where they will cut. Different enzymes will cut at different patterns. So that way you can find the exact gene you wish to use. So then the gene and the plasmid are combined together. This pattern acts like a sticky end, and it sticks the DNA back together at that point, creating recombinant DNA. It's been recombined from two different pieces of DNA. Finally, let's look at the whole process again one more time, but adding a little more detail. Remember, we remove a plasmid from a bacteria. Then we remove a gene from the foreign cell that we wish to use. Plus, not only is the gene we want on there, but we also add a gene for antibiotic resistance. This way, we can tell if the plasmid took up that DNA that we want it to. If it took up, say, the insulin-producing gene, it will also take up the antibiotic resistance gene. Once the plasmid is inserted back into the bacteria, we use an antibiotic on the bacteria, and if the bacteria is resistant to the antibiotic and it lives, then we know it picked up the antibiotic resistance gene, and it also picked up the insulin-producing gene or whatever. So it's a pretty cool way to tell if your experiment worked. So the genes are removed, You cut the gene with enzymes, let them react together. Now, when you put your plasmid and your gene together, sometimes two plasmids react. Sometimes two of the genes react, and sometimes the plasmid and the gene react the way they're supposed to. So you've got two bad and one good options that can come out of this mixture. When you cause the bacteria to take up this plasmid, it may also take up one of the not so good ones. So we only want the bacteria that took up the good plasmid to react. We don't want it if it got two plasmids. We don't want it if it got this. If that antibiotic resistant gene gets taken up, then when we treat this bacteria with antibiotics, this one will not die. This one will die and this one will, but not the one that we want. That's the one that will continue to multiply and grow, and then we can take that bacteria and just multiply it and grow it until we have as much as we need. This process is actually easy enough that it can be done in a high school science laboratory. Isn't that crazy? We have actually done this in our classroom in the past. We took some harmless bacteria and a glow-in-the-dark jellyfish gene, just like they put in those fish at Walmart, and we put that together, put that back in the bacteria, and we actually created glow-in-the-dark bacteria in our lab. Can you imagine? There's got to be some people at home working on stuff like this in their garage. Who knows what experiments are going on right now? Kind of crazy, huh?